Good afternoon, everybody. Thank you very much for the opportunity to present our work here. First of all, I would like to thank the help of my colleagues and also the long-lasting support of my mentors, Miklós Kellerman and Balázs Sarkadi. Today I will talk about a topic which was done in collaboration mostly with Gergely Lukács, leading scientists in the field working in Canada. They are doing experiments and we are doing in silico work, so I'm also very thankful not only for the hardware but also for the intellectual help of IT people and physicists. So in the last decades we were working on ABC, ATP binding cassette transmembrane proteins. They are sitting in the plasma membrane, possessing two large transmembrane domains and two nucleotide binding domains in the cytosol. They bind ATP at the nucleotide binding domain and they use the energy of the binding and hydrolysis to transport some substrate from the cell to the outside. This is true for the human proteins and most of the human proteins. We are interested in a special subgroup of these proteins which recognize a very wide range of chemically different compounds. They transport xenobiotics, toxic molecules out from the cells, keeping their concentration in the cell low. These proteins are connected, causes multi-drug resistance of cancer cells against the chemotherapeutic treatment, and they also alter the pharmacokinetics of any kind of drugs like antidepressants, etc. But today I will talk about ABC protein, which is not an active transporter, it is a chloride channel, and use the energy of ATP binding and hydrolysis to open the chloride channel. And the chloride will flow according to the electrochemical gradient. Mutations in this protein called CFTR cause cystic fibrosis, which is a monogenic disease with high mortality and morbidity. Because it is a chloride channel, the mutant protein resulted in imbalanced salt and water homeostasis. Although the gene was cloned almost 30 years ago, and we know over 2,000 mutations, till now we don't have any real cure for the most frequent mutation, which is present over 80% of the patients. It is called delta F508, which is the deletion of the phenylalanine at the position 508. The CFTR protein is synthesized in the endoplasmatic reticulum, similar to any other transmembrane proteins. When it is correctly folded, it is transported to the Golgi compartment where it gets glycosylated and routed to the plasma membrane where it functions. However, the mutant forms cannot fold correctly at the ER, so they are not transported further, but they routed to the proteasome for degradation. So the main goal in the field to develop drugs which corrects the folding of mutant proteins, especially the delta F508. Currently there are a few drugs under clinical investigation and also some approved, such as umacafrator, but they are not effective at all, not really effective. Very, very marginal effects can be observed. So in order to develop much better drugs, you need a structural understanding how this mutation disturbs the protein structure at the atomic level. The high-resolution structure of CFTR was published just a few months ago. Here you see the transmembrane domains labeled by green colors and the nucleotide binding domains with orange and red. And here, which I magnify, you see the phenylalanine 508 with black and its connection to the transmembrane region, and this small region is highlighted with blue. So if this phenylalanine is deleted, the domain-domain interactions are affected. Gergely Lukács group has shown this more than 10 years ago, and we also described this structural interaction without the structure, but using a homology model. And it seems that most of the drugs nowadays interact in this region and correct this domain-domain interaction. But they are not effective since this is a secondary cause only, or a second step problem, since the folding and stability of this NBD, nucleotide binding domain, is already affected by this mutation. My collaborators investigate the folding stability of this domain by experimental method, for example, like the hydrogen deuterium exchange method. If you incubate the isolated domain in heavy water, then the amide hydrogen can be replaced with deuterium if a given region is very mobile. And after a certain time you stop the reaction, you digest the protein and determine the peptide fragments and also the level of deuteriation by mass spectrometry. And as you see, you can detect deuteriation, so dynamics of certain regions, which are approximately 10-15 amino acid long regions. So the resolution of this method is relatively small for drug development. As you see, just conclude these experiments, that compared to the wild type, the blue lines, the mutant NBD1 in all regions are much more mobile, more susceptible to hydrogen deuterium exchange. In order to get an atomic level description of the events, we do in silico computational experiments. In order to study or to compare the wild type and the mutant protein, we perform molecular dynamics simulations. Molecular dynamics simulations solve the Newton's equations of motions, and the forces are described by interactions between atoms, determined by empiric, semi-empiric, so-called force fields. However, if we perform molecular dynamics simulations, our time scale, what we can describe, is relatively short at the nanosecond time scale. However, proteins moves, biological important movements happens at the millisecond time scale. The second problem is if you see the motions in the wild type and in the mutant protein, it's very difficult to dissect what is the difference between the two simulations. So at this moment, people use network-based analysis of motions. We try to correlate motions in the protein. We determine correlations in motions between all amino acid pairs. We build a graph and analyze the graph. So the nodes of the graphs are amino acids, and we define edges between pairs if amino acids are contact over 75% of the simulations. Contact means that they are closer than 10 angstroms, for example. And we weight the edges by the displacement vector correlation coefficient. And then we can perform various analyses of this graph. For example, you can perform community analysis. And here you see the result of simulation with the wild type and the mutant nucleotide binding domain. And the amino acids in a given community are colored with the same color. This means that amino acids in one community moves together. Their motions are correlated. Here you see one of the subdomains and in the wild type, and they contains two communities, the red one and the green one. In contrast, in the mutant protein, as you see, there are more communities detected, which means that the correlated motions of amino acid groups just start to get less and less correlated. It would be also very important to identify, it is very important to identify critical amino acids. For example, those amino acids which connect two communities, since those positions will be very sensitive to mutations, for example. However, this is the generally used methods for motion correlation calculations, but if the motions are close an angle 90 degree, then they can be correlated, but the correlation coefficient will be 0. So we also try to use a more robust method from Gabor Székely, which is based not on the motion but the distance. Here I show just a very, very basic formula, and it will be detailed at the end of my talks. And here I show the nodes, not by amino acids, but a larger structural segment. For example, one node is the 7th helix in the structure, or the 2nd beta strand. And I connected these nodes based on the distance correlation coefficient. And as you see, compare the mutant when is compared to the wild type, then less correlated motions can be observed. And very interesting, when we introduce so-called secondary side mutations in the mutant domain, which can correct the function of the protein, as shown by experiments, and we perform simulation with this rescued mutant protein, we can see that the motion correlations are restored. So most likely this method can be really study the correlations of motions in our system, but itself it's not enough for drug development. You really want to know the amino acid events, which amino acids are more critical. So again, I draw a different graph with the amino acids. They are connected based on the distance correlation coefficient. And for example, if I magnify this region, you see that there is just one edge between these two amino acids. So we can say that these two amino acids will be important for providing the wild type-like motions of this domain. I mean, at least contributing to the wild type-like motions. And very interesting that if we notice this 539 position is a position for a rescue mutation. So we already had the experimental data that this amino acid is important. Of course, this is our ongoing work, and we try to identify other amino acids which seems to be critical in this motion network, and we try to test them also experimentally. So that was the experiment, that was the in silico computation. Now a little bit I try to talk about GPUs. We run molecular dynamics simulations using GROMACS. Molecular dynamic simulations can be partitioned to different main tasks. One of them is to calculate the short-range non-bonded forces, particle-particle interactions. The other large portion is calculate the long-range interactions. Small part is the bonded interaction and some other jobs. And a few years ago, some part of the code was moved to GPU, and this is the particle-particle short-range interaction calculations. And all the other parts stayed at the CPU, which is not surprising since this long-range interaction calculation needs intensive inter-node communications and also involve 3D FFT calculations. So most likely you already know then it just doesn't work to go from GPU to CPU, to a CPU to another node, and then to the GPU. So therefore it stayed at the CPU. On the other hand, I think that nowadays more and more clusters has one node with a relatively high number of CPUs, like 48 CPUs. So most likely it would be worth to move also these long-range calculations to GPU if you use just one node. The other part of our study involves analysis of our trajectory. Trajectory means that we move the protein and we have snapshot every 100 picoseconds, so one frame, one structure, and somehow we want to dig some information out of this. Here you see the distance calculation formula I showed earlier, and here you see all the text from this formula, which involves a lot of matrix operations. And in my case, this small domain involves 250 amino acids, so I calculate the distance correlation 250 times 250 symmetric matrix. But what is very important that also at each point I have a vector which is the length number of my frames. So in my case it was something between 4 and 8,000. And in order to calculate this distance correlation coefficient matrix, in order to keep intermediate matrices, not to repeat certain calculations, I really needed a large amount of memory. But even if I didn't write this to the disk, it took 1 hour run. And it doesn't seem to be long, so you just simply submit the analysis and wait for it. On the other hand, if you have to analyze 20, 30, 40 simulations and you have just one computer to calculate it, this type of simulation really can be rate limiting in analysis if you have to resubmit, for example, a paper. And most likely, this calculation can be delivered to GPU, and if the whole method will be proved for us that it is worth to use these distance calculations for this type of analysis, then we most likely will try to move it to GPU. So in summary, we employ different in silico tools to understand changes at the atomic level in protein dynamics, and we also try to identify critical residues. And the objective is to develop some drugs to restore the wild type-like behavior of mutant proteins. And both the running and analysis of simulations are pretty critical for us. On the other hand, even with the cutting-edge hardware, and GROMARS is a pretty fast simulation software. The most challenging step still are the interpretation and presentation of the results because of the complexity of the data we have. I really wanted to balance a little bit better for you the subtopics of my talk. Maybe it was a little bit too much biology, so I really thank you very much for your attention.
Any questions to speak?
To be exact, I would actually have a large cluster of questions, but maybe we can discuss that a little bit later. The one that kind of popped into my mind that your method, which is, if I saw distance correlation analysis, how does that compare to checking the covariance matrix and doing principal component analysis on the contact map, for instance? Because I know that there are implementations that actually run pretty fast in that direction already.
Yes. The thing is that if I recall well, most of these correlation methods, including principal component analysis, includes the displacement vector, and I think that using the displacement vector just because of the 90-degree combination results in zero, not perfect. The other thing is most of these methods, as I mentioned, takes the physical contacts into account, and I think this is also a bad simplification. They use this simplification in order to avoid to have a very dense graph with a lot of edges. On the other hand, if you think about a long helix, and there is a correlation of movements between the amino acids between the two amino acids at the two ends of the helix. They are not in physical contact, but they are really correlated in motions because the helixes are usually relatively stiff.
Would that show up on your distance-based correlation analysis?
And I hope so, it seems.
Okay. Thank you.
Size of the FFT.
Please.
I was going to ask the size of the FFTs, like how many points would you run?
How many 50?
How many points?
Oh, no idea. I use GROMARS relatively as a big box, black box.
Okay, so if no other questions, then let's call the speaker again.